Comparing, record. Right. So this is going to be our fluid mosaic model sketchnote, and we're going to start off with titling this thing as the cell membrane. All right. So let's start off here. What is that? It is a, this whole thing?
Yeah.
It's a phospholipid. Yeah. So we're going to start off with some phospholipid structure. Okay. Phospholipids have many parts to them, but this whole top region up here we will call the hydrophilic head. Okay. And then this entire bottom region down here is the hydrophobic tails. And then we just got to fill in what they're actually made out of. Okay, so the hydrophilic head. If something is hydrophilic, is it polar or nonpolar?
Polar.
It is polar, because water is polar, and like dissolves like. So polar things can interact with other polar things. So this thing will be polar. Okay, and it is made up of a few different parts. First of all, this little R up here. Remember that R groups just mean that anything could be there. In this case, it's a choline. You don't need to know that. So I'm going to skip down to this portion right here. This is one of the functional groups that I wanted you guys to learn, and this is one of the reasons why we see it here and in ATP. So we have a phosphorus atom surrounded by 4 oxygens. What is that? That's a phosphate group. Good. Okay. And then if you go down from that, the phosphate group is attached to this 3-carbon sugar, and this 3-carbon sugar is attached to 2 fatty acids. What is this central part that's attaching everything? Starts with a glyss, ends in an rol. Yeah. Glycerol. Okay. Where else do we see glycerol? Triglycerides. Yeah. So triglycerides was a glycerol with 3 fatty acids attached to it. In a phospholipid, we see a glycerol with a phosphate group attached and then 2 fatty acids. So now we come down to the hydrophobic region down here. These hydrophobic tails are made out of 2 fatty acids. This one looks very straight and doesn't appear to have any double bonds. How would we classify this fatty acid? It is saturated, so this is a saturated fatty acid. Okay. This region right here, or on this fatty acid, we see a double bond right here, and then that's causing this kink in the chain. Okay, how would we classify that one? That is unsaturated. So it's unsaturated fatty acid. Okay. And this hydrophobic region down here, if it's hydrophobic, is it polar or nonpolar? It is nonpolar. Okay. So what we end up seeing is if you take a whole bunch of these phospholipids and throw them into a beaker of water, they would immediately wrap up in a very specific and predictable orientation based on the regions that are hydrophilic and the regions that are hydrophobic. The hydrophilic regions are going to try to interact with water. The hydrophobic regions are going to try to hide from water. So this brings us to the idea of a phospholipid bilayer, 2 layers of phospholipids. So we'll move over here now. And we will do the phospholipid bilayer section. Remember that bi just means two. Okay. Is there water inside of a cell? Yes. Is there water outside of a cell? Yes. So there would be water down here and water up here. Now remember, we're just looking at a cross-section of the phospholipid bilayer. In reality, this whole thing would wrap up and form a sphere. But we're just looking at like the side of one little chunk. Okay, so because there's water both inside and outside of the cell, we end up with this phospholipid bilayer where the hydrophilic heads, so right here we would have a hydrophilic head, are going to interact with the water on both sides. And then the hydrophobic tails, so right here, are not going to be able to interact with water. And since they don't want to interact with water, they are going to wrap up and move to the inside of the bilayer, where we have the hydrophilic heads protecting the hydrophobic tails on the inside from the water. So there's not going to be any water here in the middle. So no water in there. Okay. So that shows kind of the formation and reasoning behind the phospholipid bilayer. What questions do you have up to this point? Good so far? Okay. So now we're going to move over and show kind of how this would occur. So come over here to our droplet of water. Okay. So how would phospholipids arrange themselves if you had water right here? Is the hydrophilic head or the hydrophobic tail going to be oriented towards the water? The head. Good. So we would have all of the heads being attracted to the water. With the hydrophobic tails facing out. But then what if there was water out here too? Yeah, so we would need a second layer, a bilayer. So we need a second layer with the hydrophilic heads protecting their hydrophobic tails from this water. So we would have another layer of phospholipids forming right here, facing the opposite direction. And this is how we end up with a phospholipid bilayer. I wanted to show it to you like this so that you could see that we're not dealing with just a straight line. These are always going to end up forming spheres. And again, we're only drawing in 2 dimensions, so I have a little diagram here that shows you what this would look like in 3 dimensions. Well, that could have gone better. But it's the extent of my artistic abilities there. All right. So, good so far? Okay. So, really what we end up seeing is this forming. We would have a sphere, and you can see how this starts to kind of look like a cell. If you look really closely, you can see where the hydrophilic heads are here compared to the hydrophobic tails. There are a whole bunch of them, and this does a good job of showing this in 3 dimensions. And just to be thorough, there would be water out here. There would not be any water in here. In between the hydrophilic heads. Okay. This is how you end up with a cell where you have that aqueous or water-soluble cytoplasm that's on the inside of the cell, and then the watery environment that is surrounding the cell as well. All right. Now, remember that your cell membranes are selectively permeable, meaning certain things can get in and certain things can get out, but certain things get blocked. Right? And we said before class that small nonpolar things are going to be able to pass through. So let's go ahead and add that in now. So we'll do that right here. Okay. So small nonpolar materials quickly pass through the phospholipid bilayer. On the other hand, large or polar or charged materials are blocked. By the phospholipid bilayer. Okay. So the question then becomes, how do we get these large polar charged things into and out of the cells? So like glucose and amino acids and other things that have to get in and out. And this is where we have to have a better understanding of the cell membrane. It's not just a phospholipid bilayer. There's other stuff going on. So now we're going to move to this big chart here at the bottom. Our current understanding of how the cell membrane works is called the fluid mosaic model. Fluid refers to the idea that things are able to get in and out of it. Mosaic just means that it's made up of a whole bunch of different things that are all working together, that have all come together. So the fluid mosaic model. And again, this is just our current understanding. It could be years from now that we operate under a completely different model as we get more evidence. Okay, so let's go ahead and start filling out our fluid mosaic model here. The first part, if we move down to this corner, this region right here is just showing all of the phospholipids. See how many there are? There's so many. So this is the phospholipid bilayer. One of the major components of the fluid mosaic model are membrane proteins. So there's all sorts of proteins that are either stuck into the membrane or associated with the membrane, and they're going to be in charge of moving materials in and out of the cell, or allowing a pathway for materials to get in and out of the cell, or to serve as signaling between two different cells, or between hormones in the cell. They perform a whole bunch of different tasks. But there's two major types of cell membrane proteins, and we're going to come over here to label those. There are, you're going to connect these, integral proteins. These are embedded. What does it mean to be embedded? Yeah, they're lodged into the membrane. They're actually like part of the phospholipid bilayer. They're stuck into the phospholipid bilayer. Okay. And then this one, you can see it's not actually stuck into the phospholipid bilayer. This one is going to be a peripheral. A peripheral membrane protein. And these are not embedded. They just kind of hang out around the cell membrane, either inside or outside of the cell, and they perform tasks that don't have to occur within the actual membrane itself. So they're going to be just inside or just outside of the cell membrane. A lot of times they'll be hanging out next to one of these hollow proteins that we're going to label next. Okay, so integral proteins, they come in a couple different varieties. Let's come over here now and label this one. Okay. This is a transmembrane protein. Transmembrane proteins are always going to be an integral protein. They're always lodged into the cell membrane. What is different between this transmembrane protein and then this one over here? What's different between those? Here and here. This one goes all the way through the membrane. Exactly. What does trans mean? Across. Trans means across. So a transmembrane protein is a protein that goes all the way across the membrane. Okay. So across the membrane. Okay. So, is this a transmembrane protein? Yeah, it goes all the way across. What's different between this one and this one? Yeah, this one's hollow. So this is a special type of transmembrane protein. We call it a protein channel. And this is how your cells are going to transport large or polar substances. It provides a passageway. It's like a little tunnel for them. And sometimes these will work through active transport where you're having to use ATP to power them. Sometimes it just provides like a doorway, essentially. Like, I like to think of the door as being one of these protein channels where if I were small enough, I could fit through the cracks. At the atomic level, there's tiny little cracks in that wall that things can fit through. Right? But I'm too big for that. I'm too big and I'm too charged. So I need a channel. I need a door that I can go through. So if I want to get out of the room, I have to go through the protein channel. So transmembrane proteins, they're a type of integral protein, and then protein channels are a separate type of transmembrane protein, and they let big stuff and charged stuff get through. Okay, then let's move up here. Here what we see is a transmembrane protein. You can see its little head sticking out here. So you have a transmembrane protein with a chain of stuff coming off of the top. This is called a glycoprotein. Anytime that you see the prefix glyco, you know that there's going to be a carbohydrate involved. Glycoproteins are a transmembrane protein with a carbohydrate on it. So this right here is going to be a carbohydrate. It's normally an oligosaccharide, and these are in charge of cell signaling. So they're going to be like reaching out into the extracellular space, so in the space outside of the cell, feeling for hormones or other cells, or maybe like white blood cells, depending on what it is. They're going to be feeling out into the ether and seeing what's going on outside of the cell. There is another kind of cell signaling molecule that's not attached to a protein, and that would be way over here. Right here. This is called a glycolipid. What do you think makes up a glycolipid? Yeah, a carbohydrate and a lipid. In this case, the lipid is just a phospholipid. So it is a carbohydrate plus a phospholipid. Okay. Both of these, glycolipids and glycoproteins, are in charge of cell signaling. I'm spelling this wrong. Use my oops. Oops. Okay, so both glycolipids and glycoproteins, they're in charge of cell signaling. There is one last structure in our fluid mosaic model, and it's down here. Does anyone remember what else is in here that we haven't talked about yet? Cholesterol. Yeah, we still have to talk about cholesterol. Cholesterol, if you remember from the POGL, is this massive steroid molecule that's made up of nothing but carbon and hydrogen, except for one single hydroxyl group. Okay. That one single hydroxyl group allows it to embed itself between the hydrophilic heads of the phospholipid bilayer. And then the big carbon rings come up into the hydrophobic region of the phospholipid bilayer. So this whole region here in white would be hydrophobic, and they're just carbon rings. And then there's one single hydroxyl group that would embed it right here. Okay. And again, this is called cholesterol. And it regulates membrane fluidity, meaning it regulates how fluid the membrane is, how easily substances pass through it. And it does this by regulating how much space there is between the phospholipids. Okay. So this is the overall structure of the cell membrane. It's composed primarily of the phospholipid bilayer. The phospholipid bilayer is going to create these spherical shapes whenever they're placed in the water to keep the hydrophobic tails away from the water while allowing the hydrophilic heads to interact with the water. And along with just phospholipids, you also have a series of proteins and cholesterol and carbohydrate cell signaling molecules that are going to add even more function to the cell membrane. And tomorrow we're going to start looking at what functions this structure allows for. All right, what questions do you have? Okay. Go ahead and stop this.